Yes. It's fine. Yeah, I haven't got checked. I mean,
All right, so good afternoon, everyone. Thanks so much for coming. So this is going to be another National Aquarium Institute seminar from our Big Dog seminar series. And today our speaker will be Dr. John Parkinson. So the National Aquarium Institute is a multidisciplinary group of people dedicated to coral reefs that can work in very diverse areas. It can be from coral, fish, sponges, microbial, genetics, and more of the social aspect. And so very kind of tackling the study of coral reefs from all sorts of different angles. And the idea is that we would be all here talking about coral reefs and from different expertise, and maybe we'll come up with better ideas of what we can do in the future, and also see how our research fits into the world research done on coral reefs. So for today, we welcome Dr. John Parkinson. He's a professor of biology at the University of South Florida. I know some of you have done undergraduates there, or were undergraduate students there. So after earning his PhD at Penn State in 2014, he completed 2 postdocs and a stint with a coral restoration nonprofit Seacore International before starting his current position. John uses molecular tools to study mechanisms that govern the establishment, maintenance, breakdown, and evolution of marine symbiosis in a changing climate. So witness on associations between corals and the microalgae. He serves on the Coral Restoration Consortium Genetics Working Group, and his projects have contributed directly to ongoing conservation and restoration efforts. He has also been heavily involved in revising coral symbiont taxonomy and systematics. John is very well published. We were just talking yesterday. Just this year, just got
great.
Yeah, it's got 2 papers in science this past year, so I think we can say it's very well published. Early career scientist, very well accomplished, and definitely a reference for all the symbiotic acid work done in the U.S. and particularly here in Florida. At least I know all of us in Florida, when we have a question about this, we go to John. So that's why I would like to have today here with us today. So before I pass it to John, I would just have a couple of housekeeping items. At the end of the seminar, there will be a question and answer session for people that are joining us virtually. They have us already on Zoom. Please type your question in the chat. For those in person, you can speak clearly so your question is picked up by the auditorium microphones, which are right above you. So the seminar will be recorded, and then we'll then send a link to all the registered participants within the next couple of days. And then for those in person, please join us at an happy hour afterwards at the Three Suns Brewery around 4:30. And that's it. So with that, go ahead, John.
Thank you. I appreciate it. Thank you guys for having me here. I recently went up for tenure, so I gave like a tenure talk, right? It's a culmination of all of your stuff you've been doing for the past 6 years. And I thought, well, do I want to give you guys my tenure talk? No, that's boring. So instead, I have switched over and I'm giving you an overview, I think, of the trials and tribulations of trying to carry a project through from the beginning to the end. It's had some major issues with this project, and hence it is titled the Little Coral Science Project It Could. And we're going to deal just briefly with COVID and with extinction and talking about some international issues that were topics. Hopefully this is going to work. Okay, so how did this whole project that I'm going to be telling you the timeline of come to begin? Well, it came from an interest on my part in coral biomarkers for the health and thermal tolerance of corals. From this, underlying desire from a lot of coral nurseries and restoration practitioners throughout the world to be able to predict how well a coral is going to perform in the future. We're dealing with climate change and a crisis for corals. We're losing corals very rapidly, and so a lot of people are interested in identifying corals that are more tolerant, propagating them, and potentially putting them back out onto the reef and having them do well. So long ago during my PhD, we started working on identifying coral biomarkers, and that sort of carries us through to today's presentation. So I promise I'll be very brief with the introduction because I assume you guys are familiar with corals, but I left it in here just in case, a little bit. And then we'll move on to biomarker challenges, that early work with gene expression biomarkers, and then some of the stuff we've been doing recently, phenotyping, predicting performance, and then trying to predict future a little bit. So yeah, corals are pretty, you can see it from space. Reefs are made up of a bunch of individual colonies. Colonies are clonal organisms, right? Or the polyps in the corals are clonal, and within them they all host symbiont cells that are photosynthetic and help them with their energy needs so that they can grow in nutrient-poor waters. We're all good here, right? Excellent. Okay, that's coral. Wonderful. This is a featured coral. Very good. For a long time. It sucks. This is the new stuff, maybe a little bit. We're interested in the evolution and adaptive capacity of coral as well, right? Some of these early models for coral death and destruction were based on coral thermal tolerance being fixed, right? But we know that corals can adapt on some level, and their symbionts can adapt too, and that whole association can adapt together. So the question becomes, can they adapt quickly enough to deal with the changes that we're throwing at them? And so you can go through these catastrophic events, see a reduction in diversity, but maybe come out on the other side with those more tolerant individuals. Those are the ones propagating to the next generation. So we're hoping that yes, we're not going to have the diversity that we've had in the past, but maybe there are individuals out there that are strong enough to persist and create future generations that will stick around even though it's getting warmer. And the hope that I have that this is possible is images like this and colonies like this, and there are a lot of examples of them there. We've got two corals sitting right next to each other. One is clearly bleached and the other one, I wouldn't say he's doing fine. He's probably stressed as well, but visually he's not bleaching. He's probably retaining symbionts. It's likely that these guys have the same symbiont species living within them. Their microhabitats are very similar, and yet there's a different outcome, and that speaks to phenotypic variation and probably underlying genetic variation. So when we see things like this, this is what natural selection acts on, right? You will be able to potentially reproduce in the future, have children, you may die, so ends your lineage of genes. They're not well suited to whatever the current environment is. So, all of my slides are going to be cut off on the top, which is where I
Maybe push that bar down to the bottom?
yeah. Yes. Is this something? Yes.
Yeah.
Perfect. That would be a lot better. Here's my email address. Okay. So I saw this photo up on the 5th floor. Look familiar? It's hanging here. Anyway, I stole this a long time ago. Point being that we have restoration efforts ongoing. We have places like nurseries where we can grow and outplant colonies. Each one of these represents a different genotype that's been clonally propagated, right? And over the past decade or so, there's been a lot of work going into figuring out the best way to do this, both in this in-water nursery and then trying to distribute the colonies to where they are needed out in the wild and try to match genotypes to environments, try to identify resilient genotypes and propagate them. So our applied question here is, can we use our genomic tools as scientists to inform this restoration practice? Or does it make more sense to just outplant everything everywhere? Collect what you can and leave it to nature. So to try and get at these questions, this coral restoration genetics working group sort of formed a while ago and involves a lot of people, some new people that aren't depicted on here. But we've met, and our overall goal is to take information that that has been published in the scientific literature and sort of translated for practitioners into concrete guidelines, like, okay, if you want to set up a nursery, here's how many genotypes of corals you should collect. Here's how far away you should travel to collect things. Here are the different types of environments, et cetera, et cetera. One of the first publications we put out was about biomarkers. And I like the example of blood pressure as a biomarker for humans because it can give us both diagnosis and prediction, right? So if you took my blood pressure right now, it would probably be slightly elevated, so I'm talking in front of a bunch of people. But if you took it, repeatedly over time and saw that it was constantly elevated, you could say, well, I'm likely to die from hypertension in the future. And then you might prescribe me certain medications to lower my overall blood pressure. And the idea is sort of similar with the coral biomarkers. Can we both identify or diagnose stress events, and can we come up with sort of an estimate of how well a coral will perform in the future? And there are lots of different things that we could do in theory if we had good biomarkers. We could monitor reef health. We could go out and see, are the corals stressed right now? If there's an explosion nearby and a bunch of pollution ends up in the water, are the corals reacting to it or not? So we can assess those acute impacts. We can identify the resilient individuals, the ones that are, not going to be hypertensive in the future. We can try to better match our colonies out plant sites and ultimately reduce the risk, effort, and cost. This all sounds great. And a lot of this type of language was ending up in, I don't want to say promotional material, but what some practitioners thought that scientists were going to be able to deliver for them. The reality is unfortunately not that great, right? This is sort of the optimistic look, but we'll get into some of the complications later on. A couple of the examples of coral biomarkers that we have right now. Color is a great one, a very simple one where you can go out and take a photograph of the coral. And if you have a record of it, you can match it up with, sort of these normalized scales for different types of corals. You can potentially track the coral's health that way. We can use things like photochemistry. Some of these satellite products like the reheating, these are all really helpful for predicting how corals may perform. Molecular stuff, so gene expression, again, proteins, metabolites, you can go in and quantify these. And if there's an association with certain, say, gene expression profiles and coral health, then you can use that to identify resilient individuals without having to take the whole colony out and put it through a stress test and then decide how it's doing. Which, to be fair, might be cheaper and easier than some of the gene expression stuff that we've done. But the idea was that if you can establish this early on, then maybe you can develop some sort of quick and easy assay to detect it in the future on a bunch of different colonies. And then last, we've got genetic things like host DNA variation, and then stuff related to their symbionts, which again would assess via genetics, which bacteria are there, which flagellates are there. All right, so again, we want to be aware of this curve. We had this excitement phase of like, yeah, biomarkers can do everything, but the reality is it's not always quite there. And I've run into a lot of challenges. Even better, look at that magic. Okay, so yeah, we just need to be careful, I think, particularly when we're interacting with, say, government agencies or especially nonprofits that may not have as much scientific expertise to overpromise and underdeliver. That's something that's happened because a lot of the biomarkers that have come out in the literature haven't ever really been applied at a wide scale. We have these issues of scaling. It's really hard to test something in just a couple of corals in the lab and then have it actually be practical for the entire population, the entire species, other species, for example. And then a lot of times these things don't really get validated all that well. I mean, we do what we can with what we have, but it often doesn't happen on a really large scale. And of course, it takes a lot of time and money. We don't have very much of either of those things in this field. So, from this, I just wanted to point out that pragmatic biomarker development is possible, but it faces a lot of challenges. Okay, one of the best cases that we could make for developing biomarkers, I think, is our Caribbean acroporid population. We have all of this big network of nurseries. We have a lot of biomass in there. A lot of them are tracked genetically, both on the host and the symbiont side. At this point, there's a lot of exchange going on. So if we did develop a biomarker, it has the best chance of doing the most good in this system. There are many resources, a few key species in Intrigue, and a broad network of researchers, practitioners, and potentially feasible implementation. So the first project I did on biomarkers was in acroporid sort of cornice here in Florida. So we're familiar with our coral nurseries. Sadly, I don't have Nova on here because when we did this in 2010, we just went to the Miami, CRF, and TNC nurseries for this particular project. But again, you guys are familiar with this stuff. The idea was to look at these well-controlled environments where all of these genotypes are brought together, the environment is very similar, to isolate the genetic effects that are driving performance differences in corals, and see if we can translate that into like gene expression variation that's quantifiable, and give us a particular profile. We identify that profile in individuals and again predict heat tolerance, and then identify resilient corals for future use. So we ended up coming down here at 4 times in a given year to try and get the crest of the heat exposure and also the cold window, because particularly in the Florida Keys, we're dealing with marginal habitat. Like it's the end of these guys' range. So you guys are even farther north. It gets pretty cold here, and that's sort of an alternative selective scenario. And so one concern as well, you can't get too heat tolerant if you have to hold back, so you're also cold tolerant. What we've learned since then is there doesn't seem to be much of a trade-off. If you're heat tolerant, you're also cold tolerant, generally speaking, or stress tolerant overall. So I did some diving, and we didn't. And I'm really throwing this up in contrast to what I'm going to be showing you in the next section of the project. I was taking these corals out of the nursery and putting them in little buckets. And in one of the buckets, I had a heater. In the other bucket, I had a heater, and I also had a bucket of ice. I would just throw the ice in the bucket whenever it got too hot. I was trying to maintain temperatures in this range to expose the corals to different heat stresses, and then measure their gene expression afterwards. This is like so terrible. This was awful. I did not have to do a good job of maintaining these temperatures where they were supposed to be, which might explain some of the variation in a minute. But again, that was the premise. And then we measured a lot of gene expression. This was before we've gotten too far into this current generation of sequencing and high-throughput stuff. So we did what was called at the time a high-throughput qPCR assay, which I don't know if they even do these anymore. But we basically got a qPCR output for every sample from every coral at every nursery across those 4 different time points. And we looked at the expression of a particular gene. We sort of picked which genes based on a preliminary RNA-seq experiment, and then we followed up with qPCR, just a subset of them. Because that was what we could afford to do. Again, not all that much money. And the more samples you want to detect the most variation, you have to make sacrifices elsewhere. So we ended up with these expression profiles. And what you're looking at is like the fold change, increase or decrease in the expression of, this is probably heat shock proteins. Under heat stress, it went up. Great. But you can see there's a lot of variation across the time points, and this is sort of one nursery, the next nursery, the next nursery. So we ended up with profiles like this for several different genes. Then we tried to correlate them with phenotypes that we thought were useful. Things like growth, mortality, and bleaching index, very crude bleaching index that we had at the time for, was the coral very good at resisting when a bleaching event happened or not? Did everybody just die or bleach? And so we tried to find associations between these expression values and the values for things like growth. And we basically looked to see, well, can you predict one from the other? And if it was strong, like, here we have a correlation of 0.75, then the colors are darker, right? And if it's weak, like here we don't have an association, then the colors are lighter. And if it's positive, it's orange, and if it's negative, it's purple. I don't want to show you one of these for each one of these corals, and for each one of the genes, we put it together in a display like this. And what you should be able to tell from this is that it's a whole lot of nothing. Right? It's sort of all over the place. It was very hard to identify expression profiles that are actually predictive of any performance measures we had. So this was just one of our early examples that developing biomarkers is going to be a challenge in this group, at least gene expression biomarkers. And so this was part of my PhD, and it's sort of why I ended up getting involved in that consortium group that I talked about earlier. All right, so there's extensive transcriptional variation and it poses a challenge to developing biomarkers. We need to think about things like seasonal effects. We have to deal with the fact that hot and cold can elicit both shared and unique responses. So again, I think there are some genes that are just stress genes and they get turned on, whether it's cold or hot or too dark out, whatever it happens to be. And then we had some weird issues with trying to decide if we wanted our biomarkers to be things that were annotated or not, because if they were annotated, then we could potentially understand the underlying biology of what was driving these differences. But practically speaking, we really just needed it to be an indicator of health. It didn't matter what was driving that pattern, right? If we couldn't annotate a gene, we didn't need to know exactly what the gene did, as long as it correlated really well with performance. Phenotyping is the most important aspect of this, and I think that's where we really also did a poor job with this initial experiment. We don't have many coral phenotypes. They're hard to measure. What exactly is heat tolerance? So we know that phenotyping is really important, but which ones do we want to assess and use as the basis for establishing our biomarkers? And so here comes our big the focus of what I want to talk about today was this NSF proposal that fortunately got funded. Great, back when you could get things funded by NSF. And Charlie Henkel really drove most of this. And the basic premise was, okay, we want to phenotype a bunch of corals and we want to look for regions of the genome that are associated with those performance phenotypes. To get at, well, okay, if we understand the regions of the genome that are important, maybe those could be targets for future, manipulation, or at least targets to identify colonies that would be useful. And again, most of this is in restoration context. I don't want to pretend that heat tolerance is the only important thing for a coral to be able to do, but the focus here is on heat tolerance. So to improve our phenotyping overall, we wanted to do a bigger experiment. We also wanted to help out with restoration at the same time. And fortunately, Ross Cunning started his job at Shed Aquarium around this time, and we got to use their boat. So cool. It's a huge boat. It's got a live well system in the back. You can take an entire colony, stick it in the back, drive it around the Caribbean. It pumps water in from the sides, so they're getting flushed all the time. You can run your experiments on the boat. You can run it overnight while you're moving between locations. There's all sorts of stuff that you can do with a boat that you can't do if you're just working from land or land-based nurseries. Nevertheless, we were still going to nurseries. And again, at this time, when we first formulated the plan, we were going to do it in the Bahamas. Okay. Cool thing about the Bahamas is it has thermal gradient, right? And the idea was, all right, let's do typical transplants. So we'll take colonies from the Bimini nursery and we'll move them to Nassau, CEI, and Cat Island. And we'll take ones from Cat Island and move them back. So we had this whole crisscross design where we went the length of, well, part of the Bahamas, and we're carrying the corals back and forth the whole time. It was awesome, beautiful, and really successful, so that's great. We managed to get everybody where they were supposed to be. And this allowed us to look at phenotypic plasticity, right? If you have the same clonal organism at 4 different locations in 4 different environments, you track it over time, you can see which differences are driven by the environment versus ones that are driven by the genotype. We can separate things that are plastic from things that are more genetically fixed. So we thought that would be really helpful for the overall goals of the project. One of the things we measured was growth rate, again, a free plastic trade. So we thought that would be helpful. Here is some data on growth rate. Basically, each one of these different quadrants is a different coral genotype. There are 2 Bimini corals at the top, 2 Nassau corals at the bottom. And then the blue highlight here is the site of origination, and then the other locations are where we transplanted the coral to. And what you're seeing is sometimes there's a home field advantage, like these Nassau corals did well in Nassau and not so well in all the other places that we took them to. Whereas with the Bimini corals, they were not doing so well in their original environment. And they actually performed better when we moved them elsewhere. And that sort of makes sense. Bimini's the hottest place. And they were basically growing under stressful conditions until we moved them elsewhere. Okay, so that was our one successful piece of data that we got out of the Bahamas before COVID hit, okay? That meant that we couldn't travel to the Bahamas anymore. It also meant that the Bahamian government used it as, there was a lot of things coalescing at this time. They ended up changing their policy towards research in the Bahamas, and they no longer made a distinction between sort of commercial research and like nonprofit research. And so if we wanted to keep working in the Bahamas, we were going to have to spend a lot of money to be able to do so, which is unfortunate. I understand why they did it, because they basically ended up someone came in and bioprospected, and then they didn't get any of the benefit out of it afterwards. They left, they sold their product, and the Bahamas got nothing. So they updated their whole system during COVID time. It made sense, but it made it really hard to work there. And we kept thinking we'd be able to go back, and we kept pushing off, calling it quits and moving on. But eventually we did. We thought, well, okay, where's an alternative location that has a big nursery network and a thermal gradient we could study? We got Florida, right? So we came back to Florida and we changed our acrobat proper Bahamian transplant project to our acroflat project, which is really sad. Ruined our acronym, whatever. The other element of this that we didn't get to do in the Bahamas, but we did get started in Florida, was using an alternative phenotyping assay to get at heat stress tolerance. In principle, it's very similar to what I was trying to do in my buckets before. We still have buckets, they're still heated up at different temperatures. But this is called the CBAS system, coral leaching automated stress system. And it just has a lot more precision and control. And what you end up doing is you fragment coral colonies and you place a clonal piece in each one of these different buckets that are heated with different thermal regime, very precise temperature control. You then PAM them, okay? Not with this PAM, that would work. We have diving PAM to go in the water. Again, you're running these guys through each bucket has a different temperature profile, but they're separated by a very small amount, just 1 degree Celsius. So you get this whole profile, and at each temperature you get PAM data, and you plot that PAM data like this. Okay, so you've got your increasing temperature treatments, and here's the performance of your symbionts, which is what you get out of anthropometry. And you're able to create this curve. And it's essentially a dose response curve, right? And the thing that we're dosing these corals with is it's like you would do this with antibiotics or something if you're trying to develop a new drug. But our dose is with heat, and we're trying to measure essentially as a comparable point for all of these different colonies, what is the temperature at which their performance of their symbionts drops by 50%? That's the effective dose 50, ED50 value. And that gives us sort of a standardized number. And it's in degrees Celsius. This coral's ED50 happens to be right here. Okay, so 35.88 degrees Celsius. You can imagine if your ED50 is higher, you're more heat tolerant. You can handle more heat. And if it's lower, then you stress earlier. This is a way to rank colonies and their thermal performance in a somewhat more precise fashion than what I was able to do before. So here's just an example of some profiles from 4 different corals. And again, here's someone who seems to be very heat tolerant. Here's someone who is relatively susceptible. If you were someone who is designing your own coral nursery, you might decide, hey, I'm going to invest a little bit more in propagating this one than I am in this one, for example. And so we did this throughout the Keys, okay, at many of the different nurseries that are around. We again took the shed boat, and because of that, we were able to assemble usually around 40 individuals, mostly in 1 or 2 days, and then we moved to the next nursery, and so on. All right, here's the outcome of this project overall. This is our 80-50 range for what was at the time all of the acroporids in the Florida Keys in the nurseries, or a good majority of them. And what we're seeing is, depending on which nursery you're at, the overall range is a little bit different, right? But there's a pretty good spread, and at most nurseries, except maybe the FWC one, we do have some individuals that seem to be very heat tolerant. Now we can average out these differences to try to kind of mean normalize a little bit. We get our overall distribution of ED50 for the corals in the nursery system. And then we just took a look at, okay, well, how are they distributed? And again, that sort of top percentile, you can find representatives across the entire region of the Florida coral reef tract, right? Some of them even came from up here. They were in a nursery. These are the original locations, the colonies, not the nursery. So that was good news, we thought at the time. No matter where you go, there are heat tolerant individuals to be isolated and brought into your nursery. You don't necessarily have to go far away, to elsewhere in the Caribbean or, you don't find these corals much beyond that, but you can stick local, and still have a lot of resistant individuals. All right, we weren't just looking at their heat tolerance phenotype. We were also collecting DNA at the time, when we were out there sampling them. And the goal here was to bring them back, these tissue samples and do some genome sequencing. And so we have shallow genome sequence data for all of the individual colonies that I showed the E50 data for. And the sort of logical thing to do when you have this type of information is a genome-wide association study. So you look for variants in the corals that are associated with their resistance levels, and lo and behold, you found the regions of the genome that are determining heat tolerance, or at least contributing to that phenotype. It's the same for looking at epidemiology data and for associations between diseases, certain genetic markers that you can test for, like 2013. Okay, so what did we find? First, we'll look at the neutral variation in these populations. Overall, these are fun graphs showing the differences in genetic diversity among different individuals. You compress all of that down to one data point, but you're basically quantifying a dissimilarity matrix between the individual coral populations and then plotting and minimizing the variation, PCA coordination. The way you interpret this is the closer 2 points are in 2D space, the more similar their genetic profiles are. And they've done a fun thing like this with humans and found that if you just look at DNA diversity and you coordinate, DNA diversity and you coordinate for individuals in Europe, you basically recreate the map of Europe because of isolation by distance. People in Spain are more likely to reproduce with each other than people in Russia, right? It's just an issue of who's where and neutral gene flow. We did the same thing here with corals, and no big surprise here. We were able to basically recapitulate the geographic distribution of the corals. So that's great. But we did have a couple of weird things, like this guy belongs down here, he's up here. How'd that happen? What was going on? So then we looked more at this structure analysis where we could analyze the basically how many lineages we have in the Caribbean in these acroporid corals, and identify things like migrants. And what this is basically telling us is these are individual coral colonies, and the bar plot is showing you what's their probability of belonging to the orange or the yellow lineage, right? These are sort of the background genetic lineages population. And what you see is that for a couple of individuals in Florida, it seems like they came from Belize or Mexico. And this adds with data that we have on the distribution of larvae throughout the Caribbean. It looks like Mexico and Belize are probably sources for places like Florida. So not a total surprise that we were able to identify some migrants. It's kind of cool, actually. The other thing you'll notice here is that there are about 5 different major lineages. One thing that we can test is the significance of that diversity. So how strong is the distinction between what the computer has basically determined as different populations? And I want to point out up here this pairwise FST value. This is the value of the differentiation between populations. If it's above about 0.15, we consider that to be a strongly significant divergent population. If it's less than that, then maybe it's just one population, essentially. And what you find, even with our graph here, the maximum value is 0.1. We never get up to what we call significant differentiation just based on this particular measure. It does vary by species, but the bottom line is these acroporid colonies throughout the Caribbean, because we pulled some samples from elsewhere in the Caribbean to include in this analysis, they're not that different from each other. It's mostly one big population. You can see that there are 5 different ones, and down here just the support showing a lot of these. It says K or number of lineages equals 1. These are just different methods of letting the computer determine based on your data how many populations it thinks there are. And for the most part, it was saying 1 or 5, and that's telling us it's one sort of well-mixed population with sort of 5 minor subdivisions. Which is also useful information, if you're trying to figure out how to set up conservation priorities and whether you can treat a coral and handle exactly the things you would want in Florida. So that's somewhat useful information. So I meant to get to the genome-wide association part. This is sort of how it works. Again, you're looking for nucleotide polymorphisms, single nucleotide polymorphisms or SNPs throughout the genome. These corals sampled. Again, we also have ED50 data for them, so we know which ones we would consider to be heat tolerant versus ones that are heat sensitive. And if you find that that SNP is highly associated with being heat tolerant rather than sensitive, then you can say, okay, this SNP is somewhere in the genome that's contributing highly to phenotype of being resistant to coral. Someone else has done an analysis like this in acroporid corals, just not in the Caribbean, over the Indo-Pacific. They also had about 200, 250 colonies. They also had a heat stress phenotype that was related to symbiotic performance. And they basically found that there were, I don't know, 2 or 3 regions of the genome that were associated with heat tolerance. What did we find? 2 or 3 spots that were associated with heat tolerance. So unfortunately, we didn't pull out a lot. And this isn't entirely surprising. Stress tolerance, A, is a probably very polygenic trait. Lots of different genes contribute to it. On top of that, we're technically just measuring symbiotic performance. So I think there is a little bit of a disconnect between how coral host is dealing with, how you measure tolerance in the host versus what the symbiont is doing and the interaction between them. So I don't have more for you today on this because it's ongoing stuff. I don't actually know myself what the identity of these regions of the genome are. It actually fall into any areas that we have specific genes that are well annotated. But it seems to be in this acroporid and also in the Pacific acroporids that it's hard to identify biomarkers based on genomic variation is the main point. Okay, so I mentioned symbionts and microbes. What do they contribute, and can we use them as biomarkers for coral health? And we know that we can do this with the symbiotic amazing, right? We know around here, hosting Dermastinium trenchi, probably a little bit more heat tolerant than a conspecific that isn't hosting that particular species, right? In this group, in the acroporids in the Caribbean, we checked everybody hosted symbiotic amphipti. So it's not in our data set. So it's not like we compare individuals that were hosting Dermastinium to ones that were hosting Symbiotidium. And that's fine. This was actually part of why it was a nice system. We didn't have to worry too much about symbiont variation. This is a simple analysis. The bottom line is all these say A3, these are all variants of the ITS2 region. They're very closely related to each other. So it's all the same species. There might be some differences within the population, but we can't really assess it based on the low, shallow genome sequencing that we did. So we're not really worried about symbiotic variation. What we wanted to measure was microbiome variation, so bacterial variation. And this is something that I hadn't done before. I'm going to close this. Get back to Mark's question later. Okay, so there are associations between certain bacteria and diseases and disease risk, for example. So we wanted to look for associations between bacteria and heat tolerance in our acroporid system. And to do that, we brought some of the same colony fragments that we had used in our experiment out in nurseries into an aquarium system. We fragmented everybody, grew them up, and we ran them through a heat stress test, like we do. And it seems my job is to mostly melt corals. We had 2 different temperature profiles, and today we're going to be focusing on what happened at our peak stress period versus what happened at the end after about 25 days of recovery. Okay, what were we measuring at this time? A couple of things. The host color score, symbiont cell density, the protein content of the hosts, symbiont chlorophyll content, photochemical efficiency, and symbiont host cell ratios. A lot of different physiological measurements. Again, we don't have too many phenotypes of corals. We were hoping that this might encompass whatever this response is, and provide a good metric by which we compare our different microbial communities to identify perhaps individual bacteria that are contributing to good performance in any one of these particular categories. All right. Ultimately, what we found was that host protein content and symbiont cell density were good predictors of whether you're going to survive. There are a bunch of different, thermal response profiles. I'm not going to get into all of that, but if you boil it down, not so surprising. the fatter a coral you are, the more reserves that you have to deal with stress at a given time. And one thing that can get corals through a bleaching event is that they have access to heterotrophy, right? The ability to capture a bunch of prey items in the water. So maybe they're not getting their nutrients from symbionts so much anymore, but if they can eat particles in the water, that can give them enough energy to get through until the symbiont population recovers. So again, and that's sort of correlated with symbiont density to an extent. So this wasn't particularly surprising, but it gave us a target to look at. One thing I wanted to point out, this is a similar draft as we've had before. We're looking at differences, this time in microbial community composition rather than in host DNA. But the idea is the closer you are, the more similar you are. And what you can tell is there's this sort of big wide circle here with all the red samples. That's when we sampled the corals right after the big heat stress. Their microbiome profiles sort of dispersed in the subordination space compared to the control in blue, which had a tighter circle. And then over time, these other circles in the background here that are also, they aren't statistically different from that blue circle. It basically tells us the coral microbiome recovered after all those days that we left them, not under heat stress. Things went back to normal for them. So this is telling us that, yeah, the microbial community could be useful as a diagnostic marker. If you've got a bunch of weird things going on in your microbiome, it's not very similar to what you have in stable conditions, that could be an indicator of stress. Not super surprising. Okay, so which microbes were important? Well, if you look at these profiles, it's just a selection of a couple of genotypes. So these are replicates of the same genotype, and each one of them is a different genotype. What you're seeing is a whole lot of blue, right? So these acroporid corals are chock full of this rickettsia aquarickettsia rora, which we think is a parasite. And the literature talks about how it might be siphoning some energy from corals. They can actually make acroporids more susceptible to disease. What we did not find in our study was any association with the amount of aquarickettsia you had and your ability to withstand heat stress. So we didn't really find any good correlations with any of our metrics here. Dark purple is how it performed if you didn't have very much aquarickettsia. Light purple is if you had a lot. I might have those backwards. It doesn't really matter because performance is pretty across the board. One thing that's kind of cool that we did pull out in terms of, okay, what are indicator taxa that if you had this particular microbe before we expose you to heat stress, you're more likely to survive at the end. A couple of things popped out. The only one that was really well annotated is this Hyphamonidacea. And this is apparently a bacteria that's associated with the skeleton and potentially with endolithic algae. So we might have been picking up on some signal that was from the skeleton, but that made these corals a bit more resilient on some level. I don't want to talk too much about how important that is because it's just very, very preliminary. And honestly, you have so many microbes, you're going to find at least one that's significantly associated with performance, right? But it was interesting that at least it was, in a category of association that we know having these guys can be helpful because they might even contribute a little bit to host metabolism. Of course, none of this really matters because all the corals died in the end, right? So we had our big green heat wave in 2023. We just outplanted a bunch of our Cercocornis and Tortugas, so we got to watch them die. It was very sad. We had our very unprecedented warming. And so even though we found a broad distribution of heat-tolerant colonies across the whole nursery range and the Keys range, unfortunately, most of them ended up expiring anyway. So we're throwing temperature assaults at them that they cannot handle at this point. So that question of whether corals can adapt fast enough to deal with climate change, it does not seem at the moment that we're making enough progress there. But I don't want to end on a total down note that one other thing that came out of doing this was we got to do a little perspective piece on policy and what we might do to combat some of these problems. There were other regions of the Caribbean that, even though they went through the same marine heat wave, it didn't get quite as bad, and they had more tolerant corals there. So we're, again, as part of this workshop group that's trying to provide advice to practitioners, including policymakers, we're trying to advocate for moving corals around a bit more. It's not as extreme as bringing them in from the Red Sea or gene splicing them. It's just trying to push natural processes to proceed in the way that they might have in the past when we had more corals. As you lose colonies, right, and they spawn, they need partners to spawn with. And if you've only got one coral left in a place that used to be a nice big reef, they don't have a chance to reproduce. So if you move corals around, then maybe you can make more self-sustaining reefs. If you bring in colonies from elsewhere in the Caribbean, sort of locally, you can potentially bring some of those heat-tolerant eels to the area. And having anything in the Florida Keys right now is better than our current situation, where everything that's out there at this point is stuff we've planted since this event. So some of the conclusions so far from this sort of long ordeal that dealt with, again, COVID, some international problems, and the end of extinction. We know that Florida's reefs are diverse, but climate change at the moment seems to be too severe for even the toughest colonies that we had on Florida's reefs. The only ones that seem to have survived were probably deeper or hidden in sort of these micro refugia. We know that our sea bass is a useful protocol for rapid and standardized phenotyping, but it does definitely have some limitations, right? I don't want to say that it's the be-all, end-all for phenotyping. It's a very specific thing. How does a coral handle being nuked over the course of a couple of hours at a really high temperature? That's not the same type of heat stress you would experience naturally out on the reef. And you're not measuring exactly the same thing. We still think it's useful for getting some initial ideas of the diversity of heat responses in your population. ED50 can potentially be diagnostic and predictive, but again, we have those caveats that I just mentioned. But if you combine ED50, genomics, and some microbiome stuff like we've tried to do in this cursed long project, we are hopeful that we might still be able to identify some biomarkers. But I still have the same reservations I brought up at the beginning. We don't have much time or money. And every time I try to do an experiment like this, we never end up with something that's like a smoking gun, like, oh, this is a really useful thing for conservation. So I don't want to pretend that we can actually intervene on behalf of corals in the way that we might have said that we could have early on in that graph of height, right? So coral biomarker development has come a long way in 10 years, but so has climate change. Challenges keep mounting, and we don't see evolution taking place in this population. But we think that some of these more aggressive interventions, like moving corals around, and not so much the acroporids, but really other species that aren't as far gone as the acroporids were before they went locally extinct. But things like orbicells, which are really important, and they've still got relatively large populations. We should probably start moving them around now to encourage some of this allele exchange. The threats that come from potentially local allele swapping, we don't think when you compare that to the damage that comes from doing nothing, we think it would be better to ultimately. Anyway, I think that's the time that I have. I want to thank all of my funders, you guys for paying attention, and of course my lab for all the work they did and the contributions they made. Most of this work was. With that, I can take questions. Someone raised their hand online first, so I want to give that a chance. Okay, well then, that's fine. Any questions here? Yeah.
Thank you for your talk. I have kind of a two-part question. So when you were doing the acro flat and you were going to all the Florida nurseries and you took like 40 samples, were those 40 assumed distinct genotypes?
Yes, they were all the same genotype.
Okay, and then do you know, because a lot of times I know the nurseries here exchange, do you know if there was one that was represented at multiple of the nurseries or even more than one genotype?
Yeah, so we were targeting genotypes that were harvested within the range of that nursery. So the nurseries do have an exchange like we did in the Bahamas, where we moved every genotype everywhere. They've pretty much done that now. But we didn't, in this experiment, we didn't target individuals that had been taken from a different nursery and put into that one.
Okay, so the ones you did were from where the nursery was established.
Yeah.
In that vicinity. Thank you.
It'd be cool to do that, so look at their plasticity, right?
Right.
But that wasn't part of this initial effort.
Thank you, John. Excellent talk. I want to ask a little bit more about the sea bass and your perspective on it. Because, so you did this experiment and went to all the nurseries and basically, said that they can stay 34, 35 degrees, like that.
Sure.
And then the bleaching event, started, everything died. So
Let me clarify again that with the sea bass, we're doing instantaneous heat stress. And it's different than a long-term heat stress. So even though the longer-term overall temperature might not have gotten as high as we hit them with, we're measuring a different response. Because we're hitting them with the high heat stress over just 3 hours. So the ED50 value isn't directly translatable how the coral would be able to handle temperatures out on the reef. It's just how they could handle temperatures if done in sea bass.
yeah,
Which gets to that issue of relevance for
yeah, the issue of relevance, like in the end, we're getting marine heat wave of several weeks or more than that. How relevant do you think the sea bass is for these scenarios? Because in the end, we go into the nurseries and we tell them, okay, you have pretty stress tolerant coral, but then we get a marine heat wave of 6, 7, 8, 10 weeks, and that's
Yeah, I think I will. So that was why I said that I think our stresses are too great. The natural stress, well, human made stresses are too great right now for any of the variation in thermal tolerance that we saw in these property columns. So even the highest ranked individuals and the lowest ranked individuals based on our sea bass didn't really matter when the real thing happened, right? So we don't really know how relevant sea bass was for this natural bleaching event. But in other natural bleaching events that aren't as extreme, they've found a good correlation between sea bass performance and bleaching tolerance in the field, or at least in some of the tests that Chris Volsberg did in the Red Sea. One thing I didn't bring up is that we had ED50 values for all of these colonies. We also had how they performed in our aquarium setting. When we tried to mimic natural conditions more, our stress was slower and it didn't go as high and it was more sustained. And so we thought, okay, this is a great test of can our ED50 perform how they did in this artificial bleaching, experimental bleaching that we did in the aquarium. And the answer to that is no. It wasn't very predictive at all. We had a lot of, the best performers did poorly, the best performers did well, the worst performers did poorly, and the worst performers did well on grab that. And we think that part of that might be related to some of our experimental issues that we have that I'm not even going to go into. But also because the acroporids, we think, have a fairly narrow, or they had a fairly narrow range of ED50 tolerance compared to other species that sea bass was initially deployed on. So you have to sort of tailor your interpretation of your sea bass population that you're measuring. This is was kind of a remnant population. It's a bunch of, these are the corals that had survived all the previous bleaching events, right? Disease events, all sorts of human feces and the stuff that we know has damaged the reef and the keys. And also frequent cold. These survivors had a relatively narrow ED50 range. And so it could just be that when we do a sea bass type experiment, we're sorting them from like 36 to 38 degrees and saying, here's a high performer, here's a low performer. But really within that, narrow window, the associations aren't that great. You have to have more extreme heat tolerance and heat susceptibility like you see in some of the other coral populations that are no longer remnant populations, right? They have the larger breadth of phenotypic variation. Ross Cunning did a study looking like this at traditional FEFM measurements versus sea bass measurements, aquarium and versus the sea bass assay, and found a really strong correlation, but only if you included the whole data set. If you cut out like the top 10% and the lower 10%, in the middle, it was still really hard to rank the colonies. So there's, your sea bass mileage may vary depending on what you're looking at.
Question. I'm physical oceanographer, just I'm amazed with all the stuff.
No worries.
In addition to hard events, there are cold events like appearance. Is it less important less stress for corals than heat waves? Or is it what?
I'm sorry, I didn't catch all of that.
Is it the cold stress that is as important as the heat
Ah.
stress where there's.
Yeah, got it. So 2010, I think well, you guys probably know better than me. 2010 was the last one I remember that was really bad cold stress year. Still, at that time, the magnitude of death was comparable to what we would get for a bleeding event. Yeah. But those cold stress events are not as frequent. Now we have more and more heat stress events occurring more and more regularly. The cold snaps seem to be less frequent. But if you look at the geological past and how these guys have been shaped evolutionarily over time, I'm guessing it's important, and there's a reason these corals haven't expanded, farther north. It has to deal partly with temperature, partly with albedo and nutrient availability, all sorts of things. But I don't know how best to quantify the relative importance of cold versus heat, but we're dealing more with heat now than cold on a regular basis.
We keep it here at the little meters for more than quarter century, ECP with temperature, in a sense of, couldn't remember any happenings.
Oh,
It looks like it's just very kind of homogeneous. It changes only during hurricanes that have appeared sometimes if it's over wind direction. Cold water, like warm, good look.
okay.
Right, regular can go.
Let me know.
Yeah, we have a lot, yeah.
You look like.
Yes, they have.
Sorry, we got some online questions.
So Brian Walker asked, are you looking to see if heat tolerance is individuals is heritable?
In this project, no. There are plenty of other studies that look at heritability of heat tolerance in different coral species, and generally found it to be high. It's obviously that's a number that varies depending on which population you're looking at. You have to interpret it within the environment they're in. But I think the usual numbers are in like the 50% range, and versus it's not super high, but it's not super low. But you get a range. But yeah, we're not doing any coral spawning stuff. I think I've done that to the extent that I can be with doing it myself, which is why I'm here with Joanna and letting her handle that part when we do the project together.
So we have a question from Grace Plans who says, hey John, great talk. What types of
Great.
research do you feel would help support HEF and transplantation work in other oceans? Some permitting agencies, do you want to see more support for these principles before introducing them?
Yeah, so the question here is about how we convince policymakers to transfer colonies across state lines, county lines, maybe national lines. And that's definitely part of the reason we wrote that policy piece was to get these conversations going a bit more. We're not providing a lot of data in support of it, but the argument is out there, and we'll hopefully generate some research, I guess, is the idea. Again, we argue a lot that doing nothing, we know what happens if we do nothing. We get the extinction of the acroporids here. So we're hoping that sort of shift in perspective might allow funding agencies and permitting agencies to allow some more of these experiments where we can move things across greater and greater distances. There have been, because the Great Barrier Reef is under one marine park authority, they've been able to do much greater distance transfers than anything we've ever proposed doing. The only thing that we've done in the Caribbean is move some corals from Honduras to Florida. Andrew Baker managed to get people to agree to that. And they're actually out there on the reef right now, and we're seeing how they do. So that's in and of itself kind of a breakthrough in this region. But in the Great Barrier Reef, they're a lot less reluctant to move their corals, across huge stretches of latitude. And they've seen heritability of heat tolerance. They've done crosses between cold water and heat water corals and found the offspring of heat tolerance. So I think when more things like that get done, hopefully it will help with the permitting here in places like Hawaii. But I know that there's also a bit of an entrenched, precautionary principle, do no harm thing. It's something, stirring this pot is something we can't undo. But I'd also argue the pot is sort of already stirred, at least in the Caribbean, it's all pretty connected. So, yeah.
And maybe one more question, we'll go back to the folks. Natalie asked, what are your thoughts on the apparently increased susceptibility to disease that some thermal stress survivors have shown?
Yeah, I'm not convinced one way or another yet if there's a trade-off between heat tolerance and disease resistance, because in some cases, and some experiments there is, and in others there aren't. So I don't have a good answer yet. I feel like the data have been kind of back and forth, and it depends on which species you're looking at, so, and it's not my field, so I'm just sort of politely stepping back from that one. It would seem to make sense that there are going to be trade-offs in these energetic processes. If you're devoting your energy towards heat tolerance, you may be more disease susceptible. But I know that there are other studies that are not finding many trade-offs when you expose them to cold, disease, heat. Basically, the people that survive well, the individuals that survive well in one stress tend to also survive well in another stress, for at least some species. So I don't know. Corals are not a monolith, right? There's a lot of diversity there. They've been diverging from each other for a very long time. So what happens in one might not be a good predictor of what happens in another. So I don't really know. Yeah.
It's a little bit an answer to one of your previous questions about what research is needed to allow this transportations between different places. In Florida, we saw that originally SWC would not allow us to bring corals from the lower keys to the upper keys and so forth. And when they changed that now allowing us to do it was when we were able to show that there was a connectivity between them, right? And so if more studies were done, like phenotyping you showed, the ones throughout the Caribbean for the acroporid was done, and actually now we have some information that even some corals from here actually come from Mexico. That would be important. Is how much? I don't think there's this type of
No, see,
things for the other species,
the
right?
problem with that is you have to do a genetic analysis and come up with a genetic management plan for every single species. It takes time, it takes money, expertise. It's a lot of work to do these things, right?
Genetically, right?
Genetically.
If you did the other types of connectivity models, that would just try, they would just.
If it's a model,
if
is
it's
it
a
real,
model,
right? I can see that being the response. I don't know. There are a couple of people who are, they want us to calculate things like, the risk of, outbreeding, depression, right? And actually come up with a number. Even performing those experiments is really hard to do. And that gets us back to the idea of time and energy. I do think personally modeling is a great tool. And if I were a policymaker, it would convince me, but
I think
some of the people who need to be convinced, they want harder data than that.
I would think that you probably would first want to go with a model like that, and then you would use just certain areas to validate it genetically. Instead of doing genetics of everything,
Yeah,
you would do the model, and at least then you could pick up some locations and do just genetics of those and see if it fits. Does the model, was it a good predictor of the genetics we're finding? Maybe
Sounds
that would
good.
be a way to shorten the
The time.
path to be able to do some action.
yeah. Good idea.
Great talk. I just have two quick questions. For the study that you show about the Caribbean and the genetic connectivity, was that done on acroporid in general, or was it specifically to cervicornis?
That was specifically cervicornis, and I'm sorry, I didn't make it clear that we, in order to make our SNP haplotype network, we had to collect colonies from elsewhere in the Caribbean, or we had other people collect colonies from elsewhere in the Caribbean, send them for that process. So that part is Caribbean wide, whereas the genome-wide association study is just for Florida, and I wasn't clear about that before.
And for the SNPs that you use, do you know what methodology you use specifically, like 30K?
No, let's put it that way. So that would be a question for Paige and Maria and Carly. They're the ones who are mostly in charge of doing that SNP panel. I know that it took them a year to settle on which SNPs they wanted to use and which ones they were confident with and with their calls and everything. So that publication will hopefully be put together over the next couple of months. But if you want a faster answer, feel free to email, well, I don't have their emails up there, but Paige definitely email me and I can put you in touch with them. If you're trying to put together your own SNP panel, I'm sure they'd be happy to help you because they're in it at the moment.
Okay, thank you.
Yeah.
Hey, John. Nice talk,
Thank you.
and good luck with your Hanger.
Thank you. It's behind me. It's either gonna work or it's not, so I don't have to worry about it anymore.
But, actually follow up with that question, I think, kind of answered it, that it's pretty impressive that you got 3 loci that might be socially heat tolerance better than nothing.
It is definitely better than nothing. Yeah,
So they're probably gonna pursue those, try to find the genes, right?
right.
The things that might be associated.
Yes, yeah, so we're gonna zero in on those regions and look at what's nearby. And by we, I mean Paige and William.
So if that's it, and there's no more questions online, so thank you, John.
Awesome, thank you guys.
The PIs are invited to come to the FD room to help a little more with John.